Good morning, everyone, and thanks to the organizers for giving me the opportunity to present our data. So what I would like to show you today is a work in which in which I will present the role of miR-214 in the interplay between tumor and stroma cells during tumor progression. I work at the Molecular Biotechnology Center in Turin, in Italy. This is the inside of our institute. Basically, can you see my screen properly?
Yes, yes.
Okay, fine. Thanks. During tumor progression, as you know, tumor cells have to move from the primary tumor mass. The primary tumor mass forms and it recruits stroma cells, okay, which then help in this dissemination. So tumor cells then detach, cross the basal membrane, and then they travel into the vessels. They intravasate, they survive in vessels, they extravasate, and then eventually they form metastasis in distant organs. So in the last few years, we had been interested in identifying microRNAs that were particularly important in this dissemination. So in the process of detachment and survival and then seeding in distant organs. And in particular, we identified two different microRNAs, miR-214 and miR-114B, which have opposite functions. miR-214 is a microRNA which promotes metastasis formation, which is strongly expressed in malignant conditions. Instead, miR-114B is poorly expressed in malignant conditions, and it protects from dissemination. We mostly study breast cancer, and we study melanoma as well. And I have to say that these two microRNAs work in a very similar manner in both kind of tumors, obviously when the tumors are strongly malignant. So what I'm going to show you here is basically the proof that this miR-214, in fact, it promotes breast cancer dissemination. So when we have triple negative breast cancer cells, human cells, which have been engineered to overexpress miR-214, and then we take these cells and we inject these cells in immunosuppressed mice. We do the same for the same cells which do not overexpress miR-214, and then we look at the formation of the primary tumors. What you can see here is that basically there is no difference in the tumor growth in both conditions. However, when we look at the dissemination here, we have a very strong dissemination for cells that do overexpress miR-214, much stronger than for control cells, as you can see here. And this is true when the cells come directly from the primary tumor. It is true when the cells have been, when the tumor cells have been injected in the tail vein. So, and this corresponds to, in fact, a very different situation at the level of cells, meaning the cells that overexpress miR-214 are cells with an altered morphology. They have a morphology which resembles much more the fibroblast instead of the epithelial cells, and these cells can move much better in terms of motility, migration. They can invade much better, they can extravasate much better. However, it's really important to underline that this microRNA does not affect the tumor growth, okay? It's only the dissemination. Now, when we take the same human cells, triple negative breast cancer cells, and we overexpress miR-114B in these cells, so the other microRNA that I mentioned before, the one that I already anticipated that it protects. Basically, when we take cells overexpressing this microRNA and we inject them in mice that are immunosuppressed, we do the same for empty cells. And we look at the primary tumor growth. What we observe is a slight decrease in the tumor growth. Okay, so here we see an effect already at the tumor growth, not much strong, but we have an effect. But more importantly, we see that the dissemination is strongly impaired. So in the lung, we find only few metastases, indicating that, in fact, this microRNA is protecting. All in vitro data go in this direction, meaning that we have an inhibition of migration, invasion, extravasation when the cells overexpress microRNA 148b, and we have a better adhesion to different extracellular matrices. Now, what I can tell you, I'm going to summarize many investigations by telling you that basically microRNA 214 and microRNA 148b work in the same axis, and microRNA 214 works upstream of microRNA 148b. And in fact, when we have a lot of microRNA 214 and we look at the level of microRNA 148b, what we can see is that the level drops. So we have an inhibition in the expression of the microRNA 148b due to the microRNA 214. And we dissected the mechanism. We know that this microRNA 214 is in fact able really to control the expression. And we know that this microRNA 148b works through various adhesion molecules. So many direct targets of microRNA 148b are adhesion molecules, which in fact worked in an important manner during the dissemination. Now, importantly here, you can see that if cells have been depleted of microRNA 214, microRNA 148b is increased. Okay, so these two microRNAs are connected. And now, if we move to human tumors and we take data sets, large data sets, 1,300 samples, for example, here, and we look at the expression of microRNA 148b and microRNA 214, what we see is that these two microRNAs anti-correlate in human breast tumors, and the same results have been obtained in metastatic melanoma. So indicating that they work together somehow. Importantly, microRNA 214 is strongly expressed, in particular in basal tumors, so in triple negative breast cancer. And here, it also predicts survival. And in fact, when patients overexpress microRNA 214, they have a very poor outcome. And in fact, microRNA 214 can be used as a biomarker in triple negative breast cancer, in triple negative breast cancers. So in summary, here we have a cartoon which is depicting the molecular mechanism of our work, showing that microRNA 214 is strongly expressed in malignant conditions, and it leads to a decreased expression of microRNA 148b, which then in turn it leads to an overexpression of adhesion molecules which are involved in the dissemination. For example, integrin alpha 5, which works on fibronectin, on extracellular matrix, or on ALCAM, which is an adhesion molecule which works in the cell-to-cell interaction, or on other adhesion molecules as well. So we could imagine to use these microRNAs as therapeutic targets. For example, we could imagine to block microRNA 214 in order to block metastasis, or to increase the level of microRNA 148b in order to block metastasis. So from a therapeutic point of view, in fact, we try different approaches. For example, here, we try to block the dissemination by using an anti-microRNA 214. So here, basically, we have mice which bear tumors formed by triple negative breast cancer human cells. And we treated these mice with an anti-microRNA 214. This is a product from Regulus Therapeutics. It's a collaboration we had with this company in San Diego. And mice that bear these tumors compared to have been treated with this anti-microRNA compared to mice that have been treated with a control molecule, basically they do not show any difference in the tumor, in the primary tumor mass. However, they show a strong difference in the dissemination, meaning the dissemination as lung metastasis is strongly decreased following the treatment, and also the circulating tumor cells are in a much lower number. So I can tell you that if we analyze the level of miR-214 in the primary tumor, this level is in fact decreased as we expected. miR-114B instead is increased, and the level of ALCAM, for example, or integrin alpha-5 is decreased. So we basically can say that an intervention like this can block the dissemination through the molecular pathway that we know. However, this kind of approach is not specific for tumor cells, meaning that this anti-miR-214 goes a bit everywhere in all the cells that can take it up. So we try then to generate some tools which work in a specific manner for tumor cells or for other cells. And what we did here, we constructed chimeric aptamers, and we used an aptamer, in this case, AXL aptamer, an aptamer which is able to bind and to recognize specifically the AXL receptor. So AXL is a tyrosine kinase receptor which is abundant in many tumor cells, many breast cancer cells, for example. And this aptamer is therefore able to recognize it and to get internalized. Now, what we did, we linked a microRNA 14AB, the protective microRNA, to this aptamer. And in our hypothesis was, in fact, that once the aptamer gets internalized, microRNA 14AB is released. So, in fact, when we went to analyze the effect of this treatment, we observed that when triple-negative breast cancer cells were treated with this compound, first of all, we observed an increased level of microRNA 14AB in these cells. That obviously these cells are cells that express a lot of AXL. And very importantly, these cells were migrating really poorly in an experiment here with transendothelial migration, in which the tumor cells had to cross a layer of endothelial cells. Okay, so indicating that in fact this compound was working as we expected. Importantly, when we took cells that did not express AXL, nothing happens. For example, the SKBR3, they do not express AXL, nothing happens, okay? Now, the cells instead that have AXL on their surface show a reduced level of ALCAM, a reduced level of alpha-5. So the pathway, the adhesion pathway is affected. More importantly, when we took mice that were bearing tumors starting from these triple-negative breast cancer cells, we treated these mice with these conjugates, so with the AXL and microRNA 14AB aptamers intratumor. So we directly treated the tumors several days a week for almost a month. What we observed was that the dissemination in the lungs was strongly decreased, and the number of circulating tumor cells was also strongly decreased, indicating that in fact this compound was working properly. So we also prepared the opposite compound, meaning we took the AXL aptamer and we linked an anti-miR-214 to it that we call the sponge here. Now, when we treat the cells with this compound and then we look at the migration, what you can see here is that in fact there is a decreased migration compared to the controls. So indicating that the depletion of miR-214, it's working. And we also know that again, the treatment works in a specific manner, because if we treat cells that do not express AXL, nothing happens. So now, at some point, we tried to understand where exactly miR-214 was distributed within the tumor mass. And we analyzed many different breast cancer tumors from data sets. We surprisingly, we found a strong link of miR-214 with the stromal signature of the tumors, much stronger compared to the tumor components. So at that point, we evaluated the distribution of these microRNA in stroma cells. And what you can see here is that, for example, when we looked at the distribution of microRNA 214 in fibroblasts, mouse embryo fibroblasts, or cancer-associated fibroblasts, or here in mesenchymal stem cells, the expression of this microRNA was really strong. So we wondered why, and we started imagining that maybe there was a passage of microRNA 214 from stroma cells to tumor cells. So making the hypothesis that the stroma cells were working as a storage cells in which there was a sort of reservoir of microRNA 214. So in order to analyze this possibility, we performed some in vivo experiments in which we used genetically modified mice. We generated mice for the overexpression of microRNA 214. This is a conditional mouse, which although at the very beginning we used the total body mouse, so meaning that we have the expression of this microRNA 214 in all the cells of the body. The expression, as you can see here, is very good. So we have mice that express microRNA 214 in different cells. These mice are, importantly, they are viable and they are fertile. So these mice can be used as recipients. So what we did, we injected syngeneic mammary tumor cells, EO771, for example, in these mice, in the fat pad, and we looked at the formation of tumor growth, comparing these mice with mice that were not genetically modified. And what you can see here is that basically there is no difference in the tumor growth. So the tumors grow equally. However, when we looked at the dissemination, we found a much stronger dissemination of tumor cells in mice that were overexpressing microRNA 214 in basically all the stroma cells. Now, we did the opposite experiment. We used also knockout mice for microRNA 214. These are also mice that are viable and fertile. So again, they can be used as recipients. So we injected, again, the mammary tumor cells in these mice. No difference between the primary tumor mass formed in these knockout mice versus wild-type mice. However, here the result regarding the dissemination was really striking, meaning that in these mice, we had very little dissemination. So indicating that the microRNA 214 in the stroma is required for the dissemination. So at that point, we tried to understand how all this was happening. We tried to understand if there was a transfer of microRNA 214 from the stroma cells to the tumor cells. And first of all, we looked at the possible presence of microRNA 214 in the microvesicles of stroma cells, for example, fibroblasts here, or mesenchymal stem cells here, mouse or human are interchangeable for all these experiments. And what you can see is that there is quite a lot of microRNA 214 in the microvesicles of all these stroma cells, indicating that microRNA 214 goes to the microvesicles. So then we try to see if, in fact, a treatment with the microvesicles coming from stroma cells was somehow affecting the capability of the tumor cells to migrate. And what you can see here is that if we take microvesicles which come from stroma cells which express a high amount of microRNA 214 and compare the results with the microvesicles coming from cells that express some level of microRNA 214, but the overexpressing ones express more. And we take these microvesicles, we treat the tumor cells, and we look at migration. We have a strong increase in migration following the treatment with microvesicles overexpressing microRNA 214. So suggesting that, in fact, there is a link. Instead, if we do the opposite experiment that you can see here, so we take microvesicles from stroma cells that have been depleted of microRNA 214, and then we look at the migration, we observe a decreased migration compared to the controls. This if we use microvesicles or if we simply use conditioned medium. However, when we take the condition medium that has been depleted of microvesicles, so in this condition medium here, there are no microvesicles anymore, basically we see no difference in the migration, suggesting that the basis of all this are really the microvesicles. And specifically, it is the microRNA-214, which is present in the microvesicles, that does the job. So our results basically suggest that there is a transfer of microRNA-214 from stroma cells to tumor cells. And this transfer affects, this we know, we have many data on that. I cannot go into the details now, but this transfer affects the entire adhesion pathway. In fact, it affects the levels of microRNA-148b, it affects the level of ALCAM, of integrin alpha-5. What you can see here, for example, is the expression of ALCAM and integrin alpha-5 following the treatment of tumor cells by using condition medium originated from fibroblast, which overexpress microRNA, in which the microRNA-214 is not expressed anymore, okay? So basically we are able to control this adhesion pathway by modulating the microRNA-214 and just using the microvesicles. Here we have an in vivo experiment in which we use the knockout mice for the microRNA-214. And what we did here, we injected tumor cells that had been treated with stroma cells rich in microRNA-214 or not. And later on, we looked at dissemination. And what you can see is that basically there is certainly an increased dissemination once the cells have been treated with these microvesicles. So to conclude, I can say that we believe that there is a transfer of microRNA-214 from stroma cells to tumor cells via the microvesicles. Once the microvesicles enter the tumor cells, microRNA-214 is released and it affects the entire adhesion pathway, which goes via microRNA-148b and which goes via adhesion molecules such as ALCAM, such as integrin alpha-5. Therefore, we aim at developing therapies based on the ablation of microRNA-214 or the increase of microRNA-214 of microRNA-148b, not only in tumor cells, but also in stroma cells. So we really need to hit not only the tumor cells, but we need to hit also the stroma cells. And we can do this by using, for example, aptamers that can specifically bind the tumor cells or the stroma cells. So I think I concluded my talk at this point. This is our town, Torino. This is our group. I really need to acknowledge the people that did the work. In particular, I want to acknowledge Francesca Orso and Lorena Quirico, the postdocs in the lab. Federico Virga, a PhD student in the lab that carry out these experiments very well during the years, helped by master students. I need to thank the previous lab members that set up the basis for all this, and many different collaborators and different funding agencies. Thank you very much. And if you have